In 2022, a team of geneticists published a paper in Nature Genetics that quietly overturned an assumption most people, including many scientists, had been carrying around for decades without ever questioning it. The study analyzed three-dimensional facial scans from thousands of people of East Asian ancestry, identified 244 gene variants across 166 genomic regions tied to facial shape, more than 60 of them never documented before, and then compared the results directly against equivalent data from European populations. For more than a century, the framing around this particular trait had been quietly one-sided. Textbooks, forensic anthropology courses, and even popular science coverage tended to treat the narrow European nose as the neutral baseline and ask why other populations diverged away from it, as if flatness itself were the thing that needed explaining. Everyone assumed the interesting evolutionary story would be why East Asian populations evolved flatter, lower-bridged noses. That assumption turned out to be backwards. When the researchers ran their evolutionary analysis, the difference in nose shape between the two groups traced back mainly to directional selection acting on Europeans, not on East Asians. In plain terms, the flatter nasal bridge may be closer to the ancestral default human condition, and the narrow high-bridged nose associated with European ancestry is the trait that was actively pushed in a specific direction by evolutionary pressure. If you have a flat, low-bridged nose, a nose where the bridge sits close to the face rather than rising sharply between the eyes, you are not looking at some evolutionary shortcut or an underdeveloped version of a standard nose. You may be looking at something much closer to the original blueprint, carried forward relatively unchanged through tens of thousands of years, while other populations around the world diverged away from it. And if you carry East Asian, Southeast Asian, or Indigenous American ancestry, this story runs directly through your genome, because the genetic architecture behind it has only been fully mapped in the last few years, and it connects you to some of the oldest continuously traceable human lineages anywhere on Earth. Hit subscribe, because every week we take a deep dive into DNA research, ancestry mysteries, and the science that is only now starting to explain where our physical features actually come from, using real, peer-reviewed studies rather than guesswork or old assumptions. To understand where this trait actually comes from, we need to go back to the same foundational research that cracked open the genetics of nose shape in the first place. In May of 2016, geneticist Andres Ruiz Linares and his team at University College London published a genome-wide study in Nature Communications using the Candela cohort, more than 6,000 genetic profiles collected across Latin America. They isolated 4 genes tied directly to nose morphology. DCHS2, a gene involved in cartilage growth, turned out to be the strongest predictor of how far the nose tip projects outward from the face and how sharply angled that tip appears. RUNX2 controlled the width of the bony nasal bridge. GLI3 and PAX1 shaped nostril width. But there was a fifth piece of the puzzle that the Candela study confirmed rather than discovered, because 2 earlier genome studies conducted specifically in European populations, published years before the Candela project began, had already linked a gene called PAX3 to something called the nation, the precise point at the top of the nasal bridge where it meets the forehead. Those 2 independent European studies didn't agree on much else, but they converged on that one specific finding, which is exactly the kind of cross-study replication that gives geneticists real confidence a signal is genuine rather than statistical noise. The depth or shallowness of the nation is, more than almost any other single measurement, what determines whether a nose reads as flat or as sharply defined in profile. A deep nation pulls the bridge upward and creates that high-bridged projecting look. A shallow nation leaves the bridge sitting closer to the surface of the face, producing the flatter profile carried by a huge share of the world's population. For years after that, the genetic map of flat and low-bridged noses specifically remained thinner than the map for width or pointiness, mostly because so much of the earlier facial genetics research had been conducted in European or Latin American cohorts, leaving a genuinely large gap in the scientific literature for the population where this trait is most common. That changed in 2021 when a team of researchers published a genome-wide association study in the Journal of Genetics and Genomics, analyzing 3-dimensional facial scans from more than 2,600 Han Chinese individuals, the largest study of its kind ever conducted specifically in that population. They found significant genetic signals in 5 separate genomic regions tied to nose and eye shape. One sat near HXD1 and MTX2 on chromosome 2, tied to changes around the nasal root. Another sat inside a gene called VPS13B on chromosome 8. A third sat right next to DCHS2 itself, confirming and extending what the Candela study had found years earlier, this time specifically affecting nose prominence. A fourth region, near PISRT1 on chromosome 3, was linked to eye shape, and a fifth, near 2 genes called SOX9 and KSK17, was tied specifically to the salience, or lack of salience, of the nasal tip. When the researchers visualized how each of these variants physically altered the face, comparing average facial scans between people carrying different genotypes side by side, they found consistent effects on 3 specific traits: how prominently the nose projects from the face, how deep or shallow the nation sits, and how the nostrils are shaped. They also went back and checked earlier findings from European studies against their own data and found that a variant near PAX3, the same gene tied to nation depth in the original European research, showed up again in their Chinese cohort, this time specifically associated with the nose rather than other facial regions. In other words, the same gene keeps appearing across completely different populations, tied to the exact same trait, which is about as strong a signal as genetics can offer that this particular piece of facial architecture is genuinely, deeply heritable, rather than a random accident of any one population's history. There's one more gene that constantly comes up in any conversation about East Asian and Indigenous American facial features, and its story is stranger and older than almost anything else in this entire field. It's called EDAR, and the specific version of it responsible for so much of this variation is a single mutation known as EDAR 370A. This gene isn't primarily a nose gene at all. It's part of a signaling pathway that controls the development of hair follicles, teeth, sweat glands, and other structures that all originate from the same layer of tissue in a developing embryo called the ectoderm. The 370A variant is associated with thicker, straighter hair, increased sweat gland density, distinctively shaped tooth enamel known as shovel-shaped incisors, a particular triangular earlobe shape, and, in the original 2016 Candela study, reduced chin protrusion. Later research across multiple East Asian populations, including studies conducted on Uyghur populations in Western China, confirmed that this same variant has a genuinely wide-reaching set of effects across facial development generally, describing it as having pleiotropic effects, meaning one single gene mutation influencing many seemingly unrelated traits at once, because they all share the same underlying developmental pathway. What makes EDAR 370A remarkable is not just what it does, but when and where it appeared. Genetic dating places the origin of this mutation at roughly 30,000 years ago, and researchers studying its spread found that it was driven to extremely high frequency through strong positive natural selection sometime before 10,000 years ago, one of the clearest and most rapid signals of recent human adaptation ever documented in any population. Today it's carried by roughly 93% of Han Chinese people, and the majority of people across nearby East Asian populations, along with a large share of indigenous peoples across the Americas, which makes sense given that the ancestors of Indigenous Americans descended from populations that had already been living in Northeast Asia and Siberia for thousands of years before crossing into the Western Hemisphere. One of the leading hypotheses for why this mutation spread so aggressively involves a small, isolated population surviving through the depths of the last ice age in Beringia, the now submerged land bridge connecting Siberia to Alaska, where researchers suspect the gene's role in sweat gland density and possibly breast milk composition may have offered a survival advantage in an environment with limited sunlight and severe vitamin D scarcity. That explanation remains a hypothesis rather than settled fact, and scientists are still working out exactly how much of EDR's broader facial influence, including any effect on the midface and nasal regions specifically, is a direct target of selection versus a side effect riding along on genes selected for entirely different reasons. To understand why flat, low-bridge noses are so deeply woven into East Asian and Indigenous American ancestry specifically, you have to go back to a population history that, until remarkably recently, was almost a complete blank in the scientific record. For decades, almost nothing was known about who actually lived in East Asia between roughly 40,000 and 9,000 years ago. Because ancient remains from that stretch of time are scarce and difficult to sequence, a gap researchers sometimes describe as one of the largest remaining blind spots in all of human population genetics. That began changing dramatically over the last several years, as ancient genome sequencing technology improved and researchers started recovering usable DNA from increasingly degraded, increasingly old remains. In 2003, archaeologists near modern Beijing discovered the partial skeleton of a man who lived roughly 40,000 years ago, now known as Tianyuan Man, whose genome remains the oldest fully sequenced ancient human genome recovered anywhere in East Asia. Genetic analysis confirmed Tianyuan Man was a distant ancestor of both modern East Asians and, through a separate and surprising genetic thread uncovered only in the last decade, some indigenous populations in the Amazon basin of South America. Evidence of a far more tangled migration history than anyone had assumed, one that suggested an unknown, unsampled ghost population, somewhere in ancient Asia, contributed DNA to both branches independently. For a long time, researchers believed the population Tianyuan Man belonged to persisted more or less continuously into the present day. Newer research overturned that too. A 2021 study identified an individual researchers call AR19K, who lived at the tail end of the last ice age, roughly 19,000 years ago in what is now the Amur River region of northeastern Asia, and found that this individual represents the earliest known ancestor specifically of northern East Asian populations, distinct from the older Tianyuan lineage, which appears to have largely vanished rather than persisted. That discovery pushed back the genetic split between northern and southern East Asian populations by roughly 10,000 years earlier than researchers had previously believed, showing that the north-south divide running through East Asian ancestry today and through the flat-nosed gene variants riding along with it is far older and far more deeply rooted than anyone expected even a few years ago. Further south, entirely separate hunter-gatherer populations, genetically distinct from both the Tianyuan and AR19K lineages, lived across what is now Laos and Malaysia, associated with an archaeological culture called Hoa Binhian. These populations eventually vanished from the modern gene pool almost entirely, replaced or absorbed by later migrations, leaving barely a trace in the DNA of people alive in the region today. A pattern of population replacement and disappearance that keeps showing up again and again throughout this deeper history, far more often than the simple linear ancestry stories most people assume. It's a useful corrective to the instinct to imagine any single population's ancestry as one unbroken thread running quietly back to the Ice Age. The real picture, revealed only through direct ancient DNA sequencing rather than assumption, is one of repeated arrivals, replacements, and disappearances, with only a fraction of the people who ever lived in a region actually leaving descendants who are still around today. The final piece of this puzzle arrived with agriculture. Around 10,000 years ago, separate groups of early farmers domesticated millet along the Yellow River in northern China, and rice along the Yangtze River farther south, two agricultural traditions that had developed largely independently of one another in different river valleys, using different crops, different tools, and different material cultures. When researchers sequenced ancient DNA from coastal populations in southern China, dating to roughly 9,000 years ago, they expected to find a population genetically distinct from the northern farmers, given how different their archaeology and material culture appeared on the surface. Instead, they found something that surprised even the researchers running the study. By 9,000 years ago, the DNA shared by nearly all present-day East Asians was already widespread across both northern and southern China, meaning the population structure underlying the modern East Asian gene pool and the flat-nosed gene variants carried within it was already largely in place before either agricultural tradition had even fully matured. From that foundation, the northern Yellow River farming populations expanded outward in multiple directions over the following millennia, carrying their DNA and the gene variants tied to nation depth and nose prominence deep into the Korean Peninsula, Japan, Mongolia, and eventually, through later migrations across the Bering Land Bridge into the Americas, where they mixed with the older AR19K descended lineages already established across Siberia and the Arctic fringe, adding yet another layer to an already deeply stacked ancestral history. This is where the story becomes genuinely more complicated than a simple climate explanation. And it's worth being precise about it because oversimplifying this specific piece is exactly how ancestry science gets distorted the more it spreads online. The same 2017 PLOS genetic study from Pennsylvania State University that tested Thomson's nose rule against real living populations, comparing nose shape across West African, South Asian, East Asian, and Northern European ancestry groups, found that most aspects of nose shape evolve essentially neutrally, shaped mostly by genetic drift, the random accumulation of small differences over time, rather than direct selective pressure. The one trait that showed a clear, statistically significant signal of climate-driven selection was nares width, the width of the nostrils themselves, which correlated strongly with temperature and humidity. Nation depth and nose prominence, the two traits most responsible for what reads visually as a flat nose, were not the traits that study identified as strongly climate-selected. That doesn't mean climate played no role at all, and the 2022 Nature Genetics study did find evidence of directional selection shaping nose differences between European and East Asian populations broadly, using a completely different statistical method built around comparing polygenic shape scores across the two populations, rather than testing individual traits, one at a time. But it does mean the honest scientific picture is more layered than flat noses evolved because of warm climates, or narrow noses evolved because of cold ones. Nation depth and bridge height appear to be driven by a genuinely complex mix of ancestral population structure, the deep, tens of thousands of years old genetic architecture inherited from populations like Tianyuan Man's descendants and the AR19K lineage, layered together with whatever more targeted selective pressure acted specifically on European populations in the other direction. Researchers are still working out exactly how much of the flat nose pattern is simply the ancestral human condition being preserved relatively intact versus an active adaptation in its own right. And that distinction matters because unchanged since deep antiquity, and specifically selected for a reason, are two very different evolutionary stories that happen to produce a similar looking result on the surface. There's a physical, functional side to this story too, one that's easy to overlook because it happens gradually over an entire lifetime rather than across generations. Facial bones don't stop changing once a person reaches adulthood, no matter what genetic variants a person happens to carry at birth. Research examining midfacial bone structure in Korean subjects found that the bones forming the nasal bridge and the surrounding maxillary region continue to shift with age, with measurable decreases in the angles around the nasal opening and the upper jaw as people get older, a pattern that appears more pronounced in men than in women according to that research. Combined with naturally higher, more prominent cheekbones, common in populations with deep East Asian ancestry, this age-related bone change can make the midface appear progressively flatter over time, independent of whatever genetic starting point a person began with. It's a useful reminder that the nose you're born with and the nose you have decades later are not always identical, shaped by both the genetic variants you inherited from populations stretching back tens of thousands of years and the slow, ongoing architectural drift of your own skeleton across your own single lifetime, two entirely different timescales operating on the same piece of cartilage and bone. Now here's where this becomes personal rather than purely academic. If you carry East Asian, Southeast Asian, or Indigenous American ancestry, and you have a nose with a low, gently sloping bridge rather than a sharp, high-set one, the genetic variants responsible for that trait trace back through one of the oldest, most continuously documented human lineages on the planet. You are connected to Tianyuan Man, whose bones sat in the ground near what is now Beijing for 40,000 years before anyone found them, and whose genetic thread reaches, improbably, all the way to the Amazon basin on the opposite side of the planet. You are connected to the AR-19K population that weathered the coldest stretch of the last ice age in the Amur River Basin 19,000 years ago, founding the northern East Asian lineage that exists today and pushing back everything scientists thought they knew about when that lineage actually split from its southern counterpart. You carry, in roughly 9 cases out of 10 if your ancestry is Han Chinese, a mutation that first appeared around 30,000 years ago and was driven to overwhelming frequency by natural selection sometime before 10,000 years ago, quite possibly inside a small, isolated population surviving one of the harshest environments human beings have ever inhabited, the frozen land bridge between Siberia and Alaska. And your ancestry connects you to the farmers who domesticated millet along the Yellow River and rice along the Yangtze, two agricultural traditions born independently of each other, populations whose genetic signature was already spreading across an entire subcontinent 9,000 years before either tradition had fully matured. That's an extraordinary amount of deep history sitting quietly in the bridge of your nose, far more continuity and far less disruption than the story behind almost any other nasal trait discussed anywhere else in this series. There's a cultural dimension worth sitting with here too, because the way this specific trait gets treated cosmetically reveals something interesting about how differently beauty standards can run in different parts of the world. In much of the West, rhinoplasty is overwhelmingly a reductive procedure, removing bone and cartilage to narrow a bridge or reshape a tip that's considered too prominent. In South Korea, which has become one of the world's leading destinations for nose surgery, drawing patients from across Asia and increasingly from Western countries as well, the entire approach runs in the opposite direction. Korean rhinoplasty is built almost entirely around augmentation rather than reduction, using silicone implants, Gore-Tex, or a patient's own rib or ear cartilage to build height and definition into a naturally lower, flatter bridge, rather than remove material from a naturally higher one. Surgeons in Seoul have developed techniques specifically adapted to this goal, from 3-dimensional custom implants tailored to an individual patient's facial proportions, to specialized tip projection methods using septal cartilage grafts, precisely because the default anatomy they're working with, the flat-bridged nose this entire story has been about, is so common across the population they serve. It's a striking mirror image of what's happening in places like Iran, where narrow noses are already the anatomical norm and people surgically pursue softness and reduction instead, or in Brazil, where a different set of ancestral nose shapes drives its own distinct cosmetic surgery culture entirely. Neither direction, augmentation or reduction, is more natural or more correct than the other. Both are simply modern cultural responses, layered on top of tens of thousands of years of population history that nobody involved consciously chose. Genetics built the canvas over tens of thousands of years of migration, isolation, and survival. Culture decides, generation by generation, what people do with that canvas, and that choice belongs entirely to the individual making it, not to any outside standard of what a normal nose is supposed to look like. The research behind all of this is advancing quickly, and it's worth remembering how recent most of it actually is. The gene most responsible for nose prominence and nasion depth wasn't confirmed in an East Asian population until 2021, The finding that European nose shape, not East Asian nose shape, may be the more actively selected trait wasn't published until 2022. The earliest ancestor of northern East Asian populations, specifically, wasn't identified until that same year, overturning assumptions about East Asian population continuity that had stood largely unchallenged for decades. This is a field where major headline rewriting discoveries are still happening on almost an annual basis, and every new ancient genome sequenced from Siberia, Mongolia, or the Amur River Basin adds another layer of resolution to a population history that was, as recently as a decade ago, understood only in the vaguest outline, more assumption and inference than actual sequenced evidence. Commercial ancestry testing companies are still catching up to this research, and reference panels built primarily from more recent population sampling don't yet fully capture the depth of lineages like Tianyuan Man's or the AR19K population, which means the ancestry percentages and trait predictions showing up on consumer DNA reports today are likely to look meaningfully more detailed and considerably more accurate within just a few years, as more of this ancient population structure gets folded into the databases those companies actually compare your genome against. If this kind of research interests you, and if you've made it this far, it clearly does, subscribe and turn on notifications, because we go deep into topics like this every single week, taking real, peer-reviewed findings and explaining what they actually mean for real people trying to understand their own ancestry. And if there's a specific ancestry question you want us to dig into next, leave it in the comments below. We read all of them, and a lot of our upcoming episodes get built directly around the questions that keep showing up. Before we close, it's worth addressing something that comes up constantly whenever this topic gets discussed because it trips people up every time. What an ancestry percentage on a DNA test actually tells you about a single visible trait like nose shape. A lot of people assume that if a test says they're mostly one ancestry with a smaller secondary component, a trait like a flat nasal bridge must obviously trace back to whichever ancestry makes up the larger share. That isn't how the underlying biology actually works. Nose shape is influenced by several separate genes, each capable of being inherited independently from different ancestral lines, recombining generation after generation in ways that make simple, single-source attribution essentially impossible. What the research actually supports is probabilistic, not deterministic. A shallow nation and a projecting nose tip reflect a specific combination of variants at genes like PAX3, DCHS2, and the region near SOX9 and CASC17. Variants that were already common in ancestral East Asian populations, tens of thousands of years before any of the migrations, farming transitions, or admixture events that followed. Every nose shape found anywhere on the planet today reflects a functional inheritance from somewhere specific in deep human history. None of them are more evolved, more advanced, or more final than any other, and the 2022 evidence for directional selection in Europeans doesn't make the flat nose primitive any more than it makes the narrow nose an improvement. They are simply the visible surface of population histories that, in the case of the flat, low-bridged nose, may stretch back further, largely unchanged, than almost any other nasal trait scientists have identified so far. A genuinely rare thing in a species whose face has been reshaped by migration and mixing again and again for tens of thousands of years.